General Biology 2, Module 1, Recombinant DNA. Most essential learning competencies are outline the processes involved in genetic engineering, discuss the applications of recombinant DNA. For learning objectives, you're going to describe recombinant DNA and genetic engineering, identify the processes involved in genetic engineering, enumerate and describe the applications of recombinant DNA, and understand the importance of knowing the pros and cons of recombinant DNA technology. Before we proceed to our lesson about recombinant DNA, let us describe what is a gene. A gene is a basic unit of heredity. It is a specific sequence of nucleotides in DNA or RNA that is located usually in a chromosome. It is a functional unit of inheritance, controlling the transmission and expression of one or more traits by specifying the structure of a particular polypeptide and especially a protein, or controlling the functions of other genetic material. What is DNA? DNA is also known as deoxyribonucleic acid. It is a large biomolecule that contains the complete genetic information for an organism. Every cell of living organisms and many viruses contains DNA. These long chains of amino acids serve as the genetic blueprints for living organisms. DNA controls how they form before birth and which traits they pass on the next generations. The basic building block of a DNA molecule is called nucleotide, and a single strand of DNA contains billions of nucleotides. Also, it composed of a 5-carbon sugar or 2-deoxyribose and a phosphate group, as well as 4 unique nucleotides that are used to create the entire DNA sequence. These are written as A, adenine, G, guanine, C, cytosine, and T, thymine. In DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. This will occur because of geometry of the base, allow hydrogen bands to form only between the pairs. As an analogous example for DNA to how the 26 letters of the alphabet can be arranged to create words with different meanings, these 4 nucleotides can be arranged in sequences to spell the genetic instructions to create all of the different protein organisms they create. What is genetic engineering? Genetic engineering is defined as the practice of purposely altering genes to achieve a specific outcome. This alteration is a modification that directly manipulates the genetic material of a living organism. Scientists can modify the DNA of bacteria, plants, and animals to add genetic information and associated characteristics from a different organism. This process is called genetic engineering, but more recently is referred to as recombinant DNA technology or genetic modification. What is recombinant DNA? It is a form of DNA constructed in the laboratory. It is generated by transferring selected pieces of DNA from one organism to another. History of recombinant DNA technology. Discovery of DNA structure, Watson and Crick in 1953. The discovery in 1953 of the double helix, also known as deoxyribonucleic acid by James Watson and Francis Crick, marked a milestone in the history of science and gave rise to modern molecular biology, which is largely concerned with understanding how genes control the chemical processes within cells. Isolation of DNA ligase in 1967. The first DNA ligase was purified and characterized in 1967 by the Gellert, Lemon, Richardson, and Herbert's laboratories. The common commercially available DNA ligases were originally discovered in bacteriophage C4, Escherichia coli, and other bacteria. Another important tool for creating recombinant DNA was the discovery in the 1960s by the Swiss microbiologist Werner Arber and American biochemist Stewart Lin that bacteria could protect themselves from attack by viruses, the productions of endonucleases known as restriction enzymes, which could seek out a single DNA sequence in a virus and cut it precisely in one place. This process prevented the replication of viruses and hence the death of virally infected bacteria. The first restriction enzyme is called Escherichia coli, was isolated and purified in 1968 by Matthew Meselson and Robert Lewin at Harvard University. These were the type 1 restriction enzymes which cleave DNA randomly away from the replication site. 2 years later, Hamilton, Smith, Thomas Kelly, and Kent Waldman at Johns Hopkins University in 1970, they isolated and characterized the first type 2 restriction enzyme, also known as KID2, from the bacterium known as Haemophilus influenzae. This type of restriction enzyme are more useful for laboratory work as they cleave DNA at the site of their recognition sequence and are the most commonly used as molecular biology tool. Jackson, Simons, and Berg, 1971, generated first recombinant DNA molecules. The first protocol for creating recombinant DNA was put forward in the early 1970s by Peter Lovan and Armin Bates-Fieser at Stanford University Medical School. In 1971, Berg, attached to Stanford University, demonstrated the feasibility of splicing and recombining genes for the first time. Jackson, Simons, and Berg, and Lovan and Fieser working on terminal transferase procedure for manufacturing cohesive ends or sticky ends of DNA molecules. 2 years later, Stanley Cohen and Herbert Boyer produced first plasmid vector capable of being replicated within a bacterial host. After Berg's 1971 landmark gene splicing experiment, the next landmark in the development of modern life technology was the insertion of recombinant DNA into bacteria in such a way that the foreign DNA would replicate naturally. This step was taken in, respectively, at Stanford University and University of California at San Francisco, successfully inserted recombinant DNA into bacteria for replication. By inventing recombinant DNA technology, Boyer and Cohen jump-started the biotechnology industry, including GeneTech, which creates important applications for a wide range of medical uses. What is recombinant DNA technology? Recombinant DNA technology comprises altering genetic material outside an organism to obtain, enhance, and desired characteristics in living organisms or active products. This technology involves the insertion of DNA fragments from a variety of sources, having a desirable gene sequence via a protein sample. One of the biological tools of recombinant DNA technology is restriction enzymes. When we say restriction enzymes, they are an important tool in genomic research. By cutting DNA at the specific site, they create a space where foreign DNA can be introduced for gene editing purposes. It is used by bacteria to destroy invading DNA. Their own DNA has been modified at the corresponding sequences they are methylates. What is meant by sticky and blank ends? When we say sticky ends, they are called as cohesive ends. They have unpaired DNA nucleotides. It is also known as overhanging single-stranded ends. While blank ends are also called as non-cohesive ends, since there is no unpaired DNA strand meeting at the end of DNA. This is also known as double-stranded non-overhanging ends. One of the examples of sticky ends is under a bacterial source known as Escherichia coli. This is called as recognition sequence of restriction endonuclease, known as palindromes. This is a sequence of units that can be read the same way in either direction. Another tool for genetic engineering is known as vectors. It is a medium by which genes of interest are transferred to hosts. There are some examples of vectors. We have the bacterial plasmid vectors. It is a small circular DNA found in bacteria. They replicate autonomously. It confers antibiotic resistance to host bacteria, allow easy identification. This is the first type of cloning vector developed. Next we have bacteriophage vectors. These have engineered phage genomes previously genetically modified platelet restriction sites. After insertion of foreign DNA, the recombinant phage genome is packaged into the capsid and used to infect host cells. Next, cosmic vectors. These have a selectable marker, a multiple cloning sites from plasmids and causites from macrophage. Artificial chromosomes. These are large fragments of DNA that can be cloned. One of the copy of such example, we have yeast artificial chromosomes and bacterial artificial chromosomes present in cells. They played an important role in the Human Genome Project. Another tool of recombinant DNA technology is a host cell. It receives a gene of interest via vector. It replicates repeatedly, passing on gene of interest to offspring. And there are two types of host cell. We have prokaryotic and eukaryotic host. And then next we have DNA ligase. It is an enzyme that creates covalent bonds, joining together a gene of interest within plasmids, producing a recombinant DNA. The gene to be inserted into the genetically modified organism must be combined with other genetic elements in order for it to work properly. So there are certain requirements of the vector. One of this is to be able to replicate in the host. This site is known as replication origin. What if I need more plasmids? This part is also called as plasmid synthesis or self-replication. Another requirement of the vector is the site for recognition of restriction endonucleases or the cloning site. It is the specific site wherein foreign DNA can be introduced for gene editing purposes. So we may insert the gene here, and how did the gene get in here? We cut the recognition sequence and paste it by means of DNA ligase at this restriction sites. Another requirement is the promoter region, or known as transcription initiation, enabled to support the gene for new DNA expression in the host. How do I know that the cells I'm growing actually contain my vector? By means of selection marker or selectable marker. It's also known as the antibacterial resistance gene. If it grows, you know. Stages of genetic engineering. The process for genetic engineering begins the same for any organisms being modified. 1. Identification of the gene of interest. When we say gene of interest, it is also referred to target gene, the particular gene being studied or manipulated in an experiment. So you're going to identify an organism that contains a desirable gene, extract the entire DNA from the organism, remove this gene from the rest of the DNA. One way to do this is by using a restriction enzyme. These enzymes search for specific nucleotide sequences called palindrome, where they will cut the DNA by breaking the bonds at this location. Isolation of the gene of interest. It means to isolate the desired DNA in pure form, free from other macromolecules. 3. Insertion of the gene to the vector molecule and ligation. Insert the new gene to an existing organism DNA. This may be achieved through a number of different processes. When modifying bacteria, the most common method is to add the isolated gene to a plasmid. This is done by cutting the plasmid with the same restriction enzyme that was used to remove the gene from the original DNA. 4. Introduction of the vector DNA to the appropriate host cell. The new gene can now be inserted into this opening in the plasmid, and the DNA can be bonded back together using another enzyme called DNA ligase. 5. Amplification of the recombinant DNA molecule in the host cell. When you say amplification, it is the process to produce multiple copies of a specific DNA sequence. Once the recombinant DNA has been built, it can be passed to the organism to be modified. If modifying bacteria, this process is quite simple. The plasmid can be easily inserted into the bacteria, where the bacteria treat as their own DNA. For plant modification, certain bacteria such as Agrobacterium chromacanthes may be used because these bacteria permit their plasmid to be passed to the plant's DNA. Recombinant DNA application. Since the first recombinant DNA molecule was created in the year 1973, the technology has been used across a wide variety of fields. Such example as modifying bacteria by adding genes that produce enzymes used in industry, like recombinant chymosin. It is also known as rennin. It is one of the main enzymes responsible for coagulation. It is found in animal rennet, and it is also used on its own for cheese making. Another is producing therapeutic products such as recombinant human insulin. Insulin from cattle and pigs was used for many years to treat diabetes and save millions of lives, but it wasn't perfect as it caused allergic reactions in many patients. The first genetically engineered synthetic human insulin was produced in 1978 using Escherichia coli bacteria. Eli Lilly went on in 1982 to sell the first commercially available biosynthetic human insulin under the brand name Humulin. It is made from recombinant DNA that is identical to human insulin. Thanks to decades of research, people with diabetes can choose from a variety of formulas and ways to make their insulin based on their personal needs and lifestyles. Insulin has come a long way. It may not be a cure for diabetes, but it's literally a lifesaver. Somatotropin is a recombinant human growth hormone used as replacement therapy in various conditions of growth failure, growth hormone deficiency, and weakness in children and adults. Recombinant Gsearch has been commercially available since 1985. Somatrem was the first available recombinant Gsearch and was largely replaced by somatotropin. Recombinant blood clotting factor 8. It is an essential blood clotting protein, also known as anti-hemophilic factor AHF. It is administered to patients with forms of the bleeding disorder hemophilia. Most people with hemophilia can use recombinant factor concentrate. Recombinant hepatitis B vaccine. It is a solution of hepatitis B sulfate antigen used to immunize patients against hepatitis B. One of the given examples in the slide is Recombinant SD, may be administered subcutaneously to persons at risk for hemorrhage following intramuscular injections. However, hepatitis B vaccines are known to result in lower antibody response when administered subcutaneously. Therefore, consider subcutaneous administration only in persons who are at risk of hemorrhage following intramuscular injections. Diagnosis of infection with HIV. The antibody test, ELISA, or Western blot, uses a recombinant HIV protein to test for the presence of antibodies that the body has produced in response to an HIV infection. The DNA test looks for the presence of HIV genetic material using reverse transcription polymerase chain reaction. Development of the RT-PCR test was made possible by the molecular cloning and sequence analysis of HIV genomes. Golden rice. This variety of rice holds substantial promise for reducing the incidence of vitamin A deficiency in the world's population. Another recombinant DNA application is for amending crops such as corn or soybean, adding pest or herbicide resistance, or increasing nutrient content. These crops are in common commercial use in several countries. Insect-resistant crops. The bacterium has been applied to crops as an insect control strategy for many years, and this practice has been widely adopted in agriculture and gardening. Recently, plants have been developed that express a recombinant form of the bacterial protein which may effectively control some insect predators. Another recombinant DNA application is recombinant vaccines for COVID-19. SARS-CoV-2, the causative agent of COVID-19, has imposed a major public health threat, which needs effective therapeutics and vaccination strategies. Several potential candidate vaccines being rapidly developed are in clinical evaluation. Health research is critical to ending the COVID-19 pandemic. Our researchers are hard at work to find vaccines and other ways to potentially prevent and treat the disease. As a result of the combined efforts of thousands of scientists around the world, a list of over 100 potential vaccines has come down to a promising few that are soon entering the large clinical trials. But what are these vaccines and how would they work? We have here the following various strategies for recombinant vaccine development. 1. DNA-based vaccine developed by cloning SARS-CoV-2 spike protein. 2. development of vaccine using DNA plasmid containing SARS-CoV-2 spike gene, and 3. Vaccine development by spike protein messenger RNA. Adam Loring, a medical doctor and a doctor of philosophy, associate professor of internal medicine and microbiology and immunology at the University of Michigan Medical School, weighed in on the leading 5 vaccine candidates for COVID-19. It describes the recent issue of the Journal of the American Medical Association. Two of the vaccine candidates, from Johnson & Johnson, Janssen Pharmaceuticals, and AstraZeneca, University of Oxford, use something called an adenovirus. What is that and why is it used? Adenoviruses are viruses that can cause the common cold. We, as humans, have them, monkeys have them, and so do other animals. For years, people have been using these viruses to deliver DNA, which are instructions for proteins. For the COVID-19 vaccine, researchers swap in a gene from SARS-CoV-2. When the vaccine is given to someone, the modified cold virus makes the SARS-CoV-2 protein, which stimulates the immune response. Another vaccine candidate from Merck, Cher, and Dome, their National AIDS Vaccine Initiative. It uses recombinant vesicular stomatitis virus. What is that? Vesicular stomatitis virus is a virus that primarily infects livestock. Cows, bulls, pigs. It is being used as a vector delivery system, much like the human or chimpanzee adenoviruses. The modified vesicular stomatitis virus delivers the instructions for the SARS-CoV-2 protein into cells. This recombinant candidate works really well against Ebola and has been proven in the field. People develop immunity to Ebola pretty quickly after getting vaccines. The final 2 vaccine candidates from Moderna and BioNTech, Pharma, Pfizer, are messenger RNA vaccines. What are those? First, it helps to remember that DNA is the key and RNA gives instructions for certain proteins. So on messenger RNA vaccine is the instructions for the SARS-CoV-2 protein. Once inside the cell, the protein is made and that triggers the immune response, just like the vector vaccine, which use viruses to deliver the protein instructions. Here you are delivering the instructions alone. It's another way of getting the protein made inside you. How will we know if the vaccines work? With most vaccines, people talk about correlative protection, what you observe, and the immune response are correlated or predict whether you will be immune or not. With most vaccines in the past, such as the vaccines for measles, hepatitis B, and seasonal flu, a good antibody response is correlated with being protected, specifically something called a neutralizing antibody response, prevents the virus from infecting the cell. There are many advantages that genetic engineering can bring the world today, and there are also several disadvantages that must be considered. Here are the following advantages of genetic engineering. And here are some of the genetic engineering disadvantages. The advantages and disadvantages of genetic engineering show us that we must carefully manage the science-based process for it to be beneficial. It is not a process that we should rush into with the hopes of quick profits or fast results. Being able to support a growing population in the human world is important. By taking a responsible approach to limit the potential for a negative outcome, we'll have the best change to have this science do amazing things for us in the future. Despite the strict regulation, genetic engineering progress, it has led to many experimental breakthroughs over the years. At the Roslin Institute in Scotland, scientists successfully cloned an exact copy of a sheep named Dolly in July 1996. This was the first successful artificial cloning of a mammal. Scientists also successfully manipulated the genetic sequence of a rat to grow a human ear on its back. These procedures are essentially a form of therapeutic cloning. Embryonic cells can now be cloned. They are grown for health purposes, such as to obtain biological organs for transplantation. Cells are also cloned in the laboratory for research purposes. How about humans? Can a human individual be cloned? At this point in time, cloning a human individual is not possible. What can be cloned is the genotype but not the phenotype itself.
part of DNA from a donor cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into it. Recombinant DNA technology involves 4 major steps. Firstly, a DNA fragment containing the gene of interest is obtained from a donor cell. Secondly, a suitable plasmid is obtained from a bacterium. Plasmids are commonly used as vectors to transfer the gene of interest into a host cell for expression. Thirdly, the DNA fragment containing the gene of interest is cut using an enzyme called restriction enzyme. This enzyme acts like scissors. It recognizes a specific base sequence and cuts the DNA at a specific point. On the other hand, the same restriction enzyme is used to cut open the plasmid. Lastly, the DNA fragment containing the gene of interest is inserted into the open plasmid with the help of another enzyme called DNA ligase. This enzyme acts like glue. It catalyzes the joining of the DNA fragment and the plasmid. A recombinant plasmid is finally formed. This recombinant plasmid can then be introduced into a host cell for different purposes, such as producing proteins of other species like human insulin, or producing genetically modified organisms that possess new characteristics.
For your group activity, you're going to use a simple paper model to simulate and learn about the process used by genetic engineers to modify bacteria. Materials to be used are the following: scissor, tape, DNA sequence for cutouts. These represent plasmid DNA and mammal DNA containing the insulin gene. From this activity, you need to print page 1 on different colored paper than page 2. And then, watch bacteria transformation as your guide. You're going to answer the following assessment questions modified as well as the guide questions. 1. Explain why bacteria are genetically modified more often than other organisms. 2. What are the risks of recombinant DNA technology? 3. Explain the importance of recombinant DNA technology applications in agriculture, healthcare, and the environment. For your individual activity, you're going to fill in the numbered boxes with the steps of bacteria transformation and answer the following guide questions. 1. What is the role of recombinant DNA in genetic engineering? 2. How recombinant DNA is formed? 3. What is the purpose of recombinant DNA technology? Why is it important? 4. What are the possible impacts of recombinant DNA technology in our world today?